Hello and welcome to the fifth lecture on this series on virology. I am Pushpangna Singh, a Prime Minister Research Fellow from IIT Kanpur. So this lecture is going to cover the structural proteins of coronaviruses, and specifically we are going to talk about the spike glycoprotein. So before we do that, I'd like to give a very small introduction on coronaviruses. So coronaviruses are enveloped, positive sense, single-stranded RNA viruses. Their genome size ranges from 26 to 32 kilobase pair, and they are the largest known among RNA viruses. There are 7 known coronaviruses which infect humans, and all these have been discussed in detail in the previous lecture covering the family Coronaviridae. So you can refer to that lecture if you want. And the size of coronaviruses range from around 80 to 150 nanometers in diameter. Now the key structural protein that we are to focus on is the spike glycoprotein, which you can see adorned on the surface of the virus. And interestingly, the spike glycoprotein is the reason why coronaviruses are named so. So early researchers, when they were looking through electron micrographs of the virus, they found that there is a halo just like the solar corona surrounding the spherical virion particle, and hence the name coronavirus came. So this envelope located spike glycoprotein is a trimer, and it is the key determinant of host tropism. So this is a basic architecture of the coronavirus genome. Again, this has been discussed in detail in the previous lecture on Coronaviridae. So as you can see here, a very small portion of the genome actually codes for structural and accessory proteins. A large portion of the genome codes for non-structural proteins, which includes polymerase and exonuclease. So I'll quickly go through the other structural proteins of coronaviruses. As I've already mentioned earlier, the spike protein recognizes and interacts with the host cell receptor. The nucleocapsid binds and protects the RNA inside the virion. The envelope protein or the E protein is a membrane channel protein, and it is involved in viral budding. The membrane protein or the M protein is responsible for packing and organizing the viral RNA. Now these four structural proteins are very crucial proteins of the coronavirus, and these to a large extent determine the infectivity of the virus and the host tropism, and the spike protein is responsible for interacting with the host cell receptor, and without its interaction with the host cell receptor, the virus cannot infect any cell. So coronaviruses are phylogenetically divided into 4 genera, that is alpha, beta, gamma and delta. This phylogenetic classification of COVs has also been discussed in the previous lecture. So this is a timeline showing the emergence of the human coronaviruses. Till now, there are only 7 known coronaviruses which can infect humans. And it all started with SARS-CoV, but the actual human infecting coronavirus dates back to something as long as 560 to 820 years ago, and that is the HCOV NL63. And after that, the HCOV 229E emerged some 200 years ago from bat alpha coronavirus, and some 100 to 120 years ago emerged the HCOV OC43, and in the early 1950s HKU1 was reported. So all these 4 human infecting coronaviruses actually cause common cold-like symptoms, and they're not that lethal. However, in 2004, in 2002-2004, the SARS-CoV epidemic was reported, and the SARS, or the severe acquired respiratory syndrome, was being caused by SARS-CoV virus. And from here started our actual struggle with diseases caused by coronaviruses. In 2012, another outbreak resulting from coronavirus, that was a MERS, or Middle East Respiratory Syndrome disease, was reported. And infections caused by MERS-CoV are so lethal that the case fatality rate is around 36%. And the COVID-19 pandemic owing to SARS-CoV-2 is what we all are dealing with right now. And like the SARS-CoV-2 coronavirus is the most infectious coronavirus to be described till date, but the MERS-CoV is the most lethal coronavirus to be described till date. For SARS-CoV-2, the case fatality rate lies at something from 2 to 5%, but for MERS-CoV, it lies at something from 34 to 36%. So coronaviruses, as you can see in this timeline here, are continuously evolving to like there's a basic tussle between trying to escape antibody neutralization and trying to enhance infectivity. So this is a basic COV entry cycle. So this cycle shows the entry and fusion and the entire replication mechanism specifically for MERS-CoV, but this is a basic outline which applies to all human infecting coronaviruses, whether it is MERS-CoV or SARS-CoV or SARS-CoV-2. So the virion first, the spike protein of the virion first interacts with the host cell receptor, and the spike protein is then activated by host cell proteases, for example TMPRSS2, and once it has interacted with the receptor, the virus can enter either through membrane fusion or through endocytosis. So these are the two basic pathways through which the virus can enter. So once it has entered either through fusing with the membrane or via endocytosis, it releases its nucleic acid inside the host cell. So the RNA genome is positive sense, and positive sense meaning this can be directly translated to produce the viral polymerase. So once the viral polymerase has been produced, RNA replication takes place and negative sense RNA genome is formed, and again the positive sense subgenomic RNA is formed, and this subgenomic RNA contains the information for encoding various other structural proteins, which are nucleocapsid, spike, membrane, and the envelope protein. Now this SEMN proteins combine with the nucleocapsid, and like the entire like once the structural proteins have been translated, they assemble in the endoplasmic reticulum, and like these MES proteins combine with nucleocapsid, and in the endoplasmic reticulum and Golgi compartment, the entire intact virion is assembled. So once the virion assembled, it gets enclosed in vesicles and it is released outside the cell through exocytosis. So our main focus in this entire entry cycle will be on the interaction of the spike protein with the receptor, because this is the interaction which forms the first line of engagement with the host cell. If this engagement is disrupted, the virus will not be able to enter inside the host cell. So understanding this interaction, understanding this mechanism, understanding the dynamics behind this interaction is very, very crucial if we want to combat deadly diseases caused by COVs. So, this is the virus surface where these red proteins are the spike proteins, and the spike interacts with the host cell receptor specifically through the receptor binding domain or the RBD. So for MERS-CoV, the receptor host cell receptor is DPP4. For SARS-CoV or SARS-CoV-2, it is ACE2. And for all coronaviruses, whether it is MERS-CoV or SARS-CoV or SARS-CoV-2, the receptor binding domain of the spike protein is the domain which interacts with the receptor. So in order for the RBD to interact with the receptor, it is very important that the key residues which can interact with the receptor to be exposed. So in the later slides, I'll discuss a very interesting phenomena which occurs, and that is the RBD breathing or the open-close dynamics of RBD. So the receptor binding domain, if it is in open conformation, the key residues are exposed and it can interact with the host cell receptor. However, if the RBD is in a closed conformation, then the key residues are not exposed and it won't be able to interact with the receptor. So this interaction of the spike with the receptor governs host tropism, and it also governs to which host the coronavirus is going to enter, to infect. So the spike protein has a glycan, this sugar glycan cap, which is present all throughout the protein. And this glycan cap also plays an important role in interaction of the spike with the receptor. So this is the reason why the spike trimer is a glycoprotein. It's not just simply a protein, it's a glycoprotein. So from here, I will discuss in detail the structure of the spike glycoprotein trimer. So this is a structure which was solved by X-ray crystallography, and this shows the pre-fusion spike glycoprotein trimer. What I mean by the pre-fusion trimer is that before fusion has happened, this is how the trimer looks like, and the structure of the post-fusion conformation is something which is different. So let's first discuss the structure of this pre-fusion trimer. So this is the this blue region is the S1 domain, and this green region is the S2 domain. So the S1 domain has a receptor binding domain or the RBD, and for MERS-CoV it is the C-terminal domain of the spike protein, and it also has the N-terminal domain or the NTD. This green domain or the S2 domain is the fusion stop. Now what differentiates this S1 domain from the S2 domain is a cleavage site between these two domains, and that cleavage is mostly governed by furin proteases. So this receptor binding domain will first interact with the receptor, and once it interacts with the receptor it remains non-covalently attached. Like after that S1 S2 cleavage, it remains non-covalently attached with the S2 subdomain. But after a second type of cleavage happens, that is S2 dash cleavage, this entire S1 cap disengages from the spike glycoprotein, and the fusion stop will have the space to open up and refold into a dramatic conformational rearrangement, and that will lead to fusion of the viral membrane and the host cell membrane. So this is the domain architecture of the spike protein of MERS-CoV, and one single protomer is around 1353 amino acids long. And as you can see here, this S1 S2 cleavage happens somewhere between here, and the S2 dash cleavage happens here. And this is the domain architecture of the spike protein of SARS-CoV-2, and a single protomer is somewhere around 1237 amino acids long. Now these are the domain architecture of single protomers, and three these kind of protomers assemble to form the spike glycoprotein trimer. As I mentioned earlier, the spike protein can exist in different kinds of conformational rearrangements and different kinds of conformational states. So this is the first conformational state, where as you can see, the RBD which is in the S1 subdomain is closed. So you can see that like in the previous slide, all the three RBDs were closed. However, here you can see this blue RBD is open, and if you compare this blue one to this yellow one and the red one, you can appreciate that the like this blue RBD is open, and the sites that can bind to the receptor are now exposed. In another structure, two of the RBDs are open and one RBD is closed. Now, these structures that you are seeing are actually cryo-EM snapshots. These can show the conformation state in which the spike glycoprotein is frozen, but these snapshots are not going to reveal what are the exact time-based dynamics of these conformational changes which are taking place. Also, another thing which needs to be mentioned here is that a state where all the three RBDs are open will be thermodynamically not a stable state, and that is why in the cryo-EM snapshots also, like this work was done by Palson et al. In 2017, and in their cryo-EM population also, they found that the population having all the three RBDs open were very less. It was somewhere around 0.03% of the entire population. So here is a video which shows RBD breathing, where you will appreciate that one RBD can open up and a second RBD will be closed and something like that. So here all the three are closed. One RBD has opened up. Second RBD has opened up. And now it is closed again. So this phenomena was termed RBD breathing by this group from Jason McClellan's lab, and this interesting work was published in PNAS in 2017. So as you can see here, there are different conformational states of not just the MERS-CoV spike but also of the SARS-CoV-2 spike, and these conformational states are the main determinants of how the spike is going to interact with the host cell receptor. Till now, the mindset in the field was that the covalent interaction of the key amino acid residues of the receptor and the spike protein specifically govern what kind of interaction is going to take place. However, with the publication of this study, it is sure that it is not just the specific sequence-based interaction, but it is also the dynamic interaction which govern host tropism. Hence, understanding the dynamic nature of these conformational states is more important than understanding just the sequence interaction of the host cell receptor and the spike protein. Now here again for the SARS-CoV-2 spike as well, it was shown that the RBD is dynamic. Like this nature is not just conserved, not just reserved to the MERS-CoV spike. It is also carried forward to the SARS-CoV-2 spike, which emerged later. So as you can see here again, like one of the receptor binding domain is in open conformation, and this shows the different angles at which, like two different angles at which the RBD is positioned, which you can appreciate from here. And it has also been shown that some mutant spikes can show different kinds of RBD movement. For example, the mutant G614G has been shown to have shown to be more prone to this RBD open conformation than the wild type spike, which is the D614 spike. So it has also been shown that binding of different kinds of antibody to the RBD or to some other regions of the spike protein can govern conformational flexibility of the spike. For example, this is the MERS-CoV spike, and for this binding of this antibody, the G4 antibody, which binds to the S2 subdomain, can govern the opening of the receptor binding domains, as you can see in red here. So it is not actually clearly understood how does the binding of an antibody in the S2 subdomain can result in a conformational change in the S1 subdomain, specifically in the RBD. Another example is the binding of a second kind of antibody, a second class of antibody, that is the G2 antibody, which binds to the N-terminal domain of the MERS-CoV spike. And once it binds to the N-terminal domain, it was shown that it leads to opening up of the receptor binding domain. And again, it was hypothesized that once it binds to the N-terminal domain, there is a steric clash because of which the RBD has to open, and once the RBD has to open, it can bind to the host cell receptor. In another kind of antibody involving the VHH55 antibody, it was shown that it can bind only when the MERS-CoV RBD is in an up conformation. A similar kind of work has also been shown for the SARS-CoV-2 spike protein. So here also, you can see that these VHs are binding to the RBDs when they are in up conformation. And there is this another different kind of antibody, which is CO2, which was there for SARS-CoV, and these can also bind to the RBD in up conformation, and their binding can induce different kinds of conformational changes in the spike protein. So all this data suggests that the conformational flexibility of the spike is not just random. It is not happening stochastically, but it is being governed by the different kind of cellular involvement that the virion is in, and the different kind of factors that can interact with the spike protein. Now, there is a second kind of conformational change which is very important for the spike protein to change from a pre-fusion conformation to post-fusion conformation and the virion to fuse. So this is the pre-fusion conformation of the spike, which shows the fusion peptide, the HR1 helix, the central helix, and the other 4 sheets. In its post-fusion conformation, you can see that there is this dramatic conformational rearrangement, and this helix has opened up. Now, once the conformation changes from the pre-fusion to the post-fusion conformation, like the helix opens up to some distance as long as 180 angstrom. Now, there has been a lot of research in trying to delineate if the binding of antibodies can affect the conformational changes in the RBD. And if they can, then how are they affecting the conformational changes in the antibody, in the spike protein? So, like as I discussed earlier, there have been a number of reports both for MERS-CoV and for SARS-CoV-2 to understand how the binding of antibody not just to the RBD but to other regions in the spike protein are affecting the conformation changes in the RBD. However, we have been limited because all the cryo-EM pictures that we get are static snapshots, and we do not get a clear idea of what is actually happening in real time and how are the dynamics being governed. So these are some of the articles that you can go through which discuss in detail uh, how the binding of antibodies to different regions of the spike are governing the conformational changes in the RBD. So this is again like the spike protein of MERS, and this binding of the G4 Fab to the S2 subdomain is affecting the open close dynamics of the RBD, as you can appreciate in the surface view here. So the first, like here, all these three RBDs are closed. Then one of the RBDs opened up, and there was another population in which two RBDs are open. And same goes for the G2 Fab, which binds to the NTD. So when it binds to the NTD, there is a steric clash, because of which the RBD has to open. In another kind of study, like it was shown that this VHH55 antibodies, which was isolated from llamas, uh, actually competes with DPP4 for binding to the RBD, and like once this antibody is present, the RBD will try to be in a down conformation, because if it is in an up conformation, this antibody can bind and neutralize the spike. So like in nature, there is a constant struggle between trying to escape antibody neutralization. Thus, the RBD has to exist in a down conformation so that it is not readily neutralized by antibodies. But if it remains in the down conformation, then it won't be able to interact with the host cell receptor, and it won't be able to infect the host. So for that, uh, it has to come up with a way in between so that it can be in an open conformation and evade neutralizing antibodies at the same time. Hence, uh, like this flexibility, this conformation flexibility exists. So membrane fusion is a very important step once the binding of spike with the receptor has happened. So fusion can happen either through fusion of the two membranes, or either by endocytosis, and which is then after which membrane fusion follows. So this is a very beautiful depiction of the viral membrane and the host cell membrane fusing, and the viral content being released inside the cell. And this painting has been done by David Goodson. So here, this is a video from Design, which shows the conformational changes of the spike which are governing membrane fusion. And here you will see the viral membrane and the host cell membrane fusing. So as you saw, like the spike completely refolded and the S1 subdomain was removed, and the S2 subdomain like went through that major conformational change of around spanning to around 180 angstrom, and the two membranes fuse, and whatever the genetic material will be in the virus will be released inside the host cell now. Now another important aspect which needs to be covered when it comes to the spike protein is the cleavage of the spike into S1 and S2 by host proteases. So there are two different kinds of cleavages which take place. The first is the S1 S2 cleavage and the second is the S2 dash cleavage. So once the virions are budding out, like this S1 S2 cleavage takes place through host cell furin proteases, and the spike glycoprotein still remains in the pre-fusion conformation. However, like when it interacts with the host cell receptor, then proteases like cathepsin and TMPRSS2 lead to this S2 dash cleavage, and this leads to this change in conformation from pre-fusion to post-fusion conformation. So this is the study which first showed that this S1 S2 cleavage S1 S2 cleavage is governed by furin proteases. However, this S2 dash cleavage is not governed by furin proteases. It is governed by specific proteases such as TMPRSS2 and cathepsin L. So this is again a remarkable study which first showed the different tectonic conformational changes of the coronavirus spike glycoprotein, and the coronavirus spike that they focused on was the HKU1 coronavirus spike, and like David Wiesler's group was the first to show that this dramatic conformational change from pre to post fusion conformation occurs. And this is the proposed model of coronavirus entry, that the spike protein first interacts with the receptor, and after that a proteolytic activation happens, and once this proteolytic activation happens, this S1 subunit is released, like it is dissociated from the S2 subunit, and once it gets dissociated from the S2 subunit, the S2 subunit gets, in very simple terms, it gets the space so that it can refold its helices, and once it refolds its helices, the viral membrane and the host cell membrane fuse. Now once we have discussed in detail the structural dynamics of the spike protein, a very important question which comes up is that, can we actually design structural dynamics correlates for the neutralization of COV infections? So what will be the implications of this? Like understanding. So what will be the implication of understanding in detail the different conformational dynamics of the spike protein, whether it is MERS-CoV or SARS-CoV or SARS-CoV-2? So the first will be structural dynamics based immunogen design, and once it has happened, different kinds of therapeutic strategies can be developed. And the most important implication will be filling the gaps that exist in understanding the entry and fusion of MERS-CoV or any other coronavirus for that matter. So there are a number of questions which need to be answered in order to reach at that stage where we can actually think of designing structural dynamic correlates for neutralization of CoV infections. So the first question which needs to be answered is, what is the real-time conformational mechanism of RBD opening and closing? Next is, what factors determine the in and out conformation of the RBD? It can either be pH, it can either be antibody, or it can just be stochastic, but the RBD movement being stochastic, the chances of RBD movement being stochastic is very little, the probability is very little, because then it will make the spike protein more susceptible to neutralizing antibodies. Like there will be 50-50 chance of the spike protein encountering neutralizing antibody or encountering the receptor. So that is something the virus will not like, because it has to make sure that encountering the receptor when the RBD is open is more probable than encountering neutralizing antibodies. The third question is, what is the mechanism of RBD rotation for a receptor accessible conformation? The fourth objective will be to test the proposed model for sequential receptor induced triggering. Proposed model is the model which I discussed in the previous slide, and like this is the model which exists in the field, where it is thought that the triggering is totally receptor induced, and the sequence of events that I discussed previously, that first the RBD opens up, then it interacts with the receptor, then the proteolytic cleavage happens, then the S1 dissociates, and then the S2 refolds. Like we do not yet know if that, like dynamically and in a real-time picture, if that is actually what it there is which is happening. So, yeah. The next question is to understand what triggers fusion. Like, what are the triggering factors which lead to fusion? What are the triggering factors which tell the spike protein that yes, the next step is to dissociate the S1 from the S2 subdomain, and the S2 subdomain to open up into that dramatic conformational rearrangement, and the two membranes to fuse. And we still do not know what are the major conformational changes involved during fusion, and what is the sequence of event of those conformational changes which are taking place in the S2 subdomain. And we know that the binding of different antibodies can affect the conformational changes of the RBD, but we do not know how does the binding of various antibodies is affecting the conformational changes. So understanding all these conformational changes in real time, in their raw dynamic nature, is important if we wish to combat, if we wish to totally overcome COV infections. Because coronaviruses have emerged to be so clever and smart that they do not only rely on sequence interaction. They are relying on this dynamic nature, and this dynamic nature is actually the nature which is helping them enhance their infectivity and evade antibody neutralization. So this is all for this lecture on the spike glycoprotein. If you have any questions or any doubts, you can shoot that up in the comment section. I hope you enjoyed the lecture. Thank you.